Thank you. So I'm Aurelia. I'm a final year PhD student here in Cambridge at the Department of Chemical Engineering. I'm supervised by Dr. David Fairen-Jimenez and by Seth here from the CCDC. And today I'll be telling you a bit about some parts of my PhD project, which consisted in building and exploring databases of porous materials for adsorption applications. So first things first, why are we interested in gas adsorption? So among the 17 global goals that were set out by the United Nations a few years ago, 3 of them are good health and well-being, affordable and clean energy, and climate action. So with adsorption, what we can do is, for instance, capture toxic compounds from the air, or store hydrogen, or capture carbon or methane, for instance, of a few examples. And the way we do this is by means of porous materials. And the idea is to physically adsorb the molecules of interest at the surface of the pores so that we can store them or separate a mixture. So when we talk about porous materials, we like to think in terms of surface area because that gives us an idea of how much adsorbate we can take. So zeolites and activated carbons are the most established materials to date, and they're widely used in the industry as well. Metal organic frameworks, or Moths, is a booming field, and it covers a wide range of surface area. And this is also the material that we are focused on in our research group. And then there's also a plethora of other porous materials with various acronyms for which there is no clear IUPAC definition, but it just shows you how active the field of porous materials is. So this talk is about building databases. So I will first tell you about the CSD Moth subset that we built a while ago. And I'm sorry if many of you have already heard this 1,000 times, but I think it just sets the background. And then I will move on to porous cages, which is the main part of my talk today. So let's start with Moths. So what is a moth? Moth is a crystal assembled, not necessarily a crystal, sorry, a structure assembled from metal clusters and organic linkers to form a framework. And there's a wide variety of clusters and linkers you can choose from. This, for instance, is Moth 5. It's one of the most famous moths. You can see the organic linkers and the metal clusters, and the sphere inside represents the cavity and gives an idea of the space. Now, as I was saying, there's a lot of different clusters and organic linkers you can choose from, and you can combine them in many ways. And so essentially designing a moth is like playing Lego. And with all the possible combinations, we can imagine that there can be a lot of structures, and maybe a lot of them have been synthesized already. And as a matter of fact, in 2017, we teamed up with the CCDC to extract all the moths from the CSD. And at the time, we found that among the 900,000 structures that were in the CSD, about 70,000 were considered as moth or moth-like. And because this subset is automatically updated, this number has reached today around 100,000, which is a huge number. And so when we have this amount of available experimental data, there are too many questions that we can try to solve. So one of them is, for instance, can we extract any knowledge from this data, and if this knowledge can guide us towards the next design of a moth. And we can also just look at the structures that are already there and wonder if the best structures are already available for whatever problem we're trying to solve. And the way that the computational moth field is tackling this nowadays is by means of computational high throughput screening. And this is the general workflow. So basically start from a huge database, say the CSD, you gather and process some data, say the CSD moth subset. You might need to clean the structures so that they're computationally usable. And then particularly in the moth field, we will move on to some geometrical characterization. And with moths, we like to use, for instance, the pole limiting diameter as a selection tool. So the PLD gives an idea of the size of the pore channel. And so we can discard some structures based on the size of the PLD because it will be too small or too large for some adsorbates. And so we end up with a selected amount of data, which is much smaller than the initial amount. And then we can move on to some more expensive molecular simulations. And using the data that we have, that we obtain from these simulations, we can then select the best structures or just study the structure property relationships of the selected data set. So this is the general idea of computational high throughput screening. But how do we actually obtain the CSD Moth subset in the first place? So Moths are extended structures, and there's a very convenient tag in the CSD that is polymeric, which targets specifically extended structures. So we use that tag as the first instance, but the problem is not all polymeric structures are Moths. Actually, it's very difficult to say what is a Moth because there is no clear definition and it's very debatable. But what we do know is that there's a wide range of structures that are definitely considered as Moths. And so we decided to focus on these structures linkages specifically. So for instance, a huge number of Moths have carboxyl ligands. And so we just designed this query to capture structures with this kind of chemical diagrams. And the Moth 5 that I showed you earlier is one of the famous examples that have carboxylate ligands. So we did the same thing with 6 other queries. And the idea is that all these criteria are integrated in the CSD so that whenever the CSD is updated, the Moth subset is also updated. And using these queries gives us a lot of flexibility because the field is always changing. So we can always fine tune them to include new types of structures that are emerging or remove structures that we've decided should not be considered as Moths or erroneous. So this is how the CSD Moth subset works. Now, I had the opportunity to work on a lot of other projects related to Moths during my PhD, but I'm not going to tell you about this today. Today, I want to focus on how I extracted cages from the CSD. And this is particularly interesting because we're basically moving from extended structures or polymeric to a set of discrete molecules. And so the first question that I had to ask myself is, what is a cage? And the IUPAC, once again, was not very useful because the definition of a cage by the IUPAC is a polycyclic compound having the shape of a cage, which, yeah, is not really helpful. But there are some things that we know about cages that can help us. So we know that there's a variety of cage shapes. So here are just some examples. They can be spherical, they can be tetrahedral, they can be helicoidal, some are more cubic. Yeah, and there's also a variety of ways of packing them. So if you look at the snapshots on the left side, you can see clear cavities. I chose them specifically because these are clear examples. And what is inside these cavities is what is called the intrinsic porosity. But then when the cages pack, they can leave some space between themselves, and that leads to some extrinsic porosity. So I guess that's what makes cages very interesting. There's 2 kinds of porosities. But actually, in order to extract the cages, we don't need to think about the packing. We can just focus on the cage shapes itself. Now, how do we actually identify the shapes of cages? Well, I came upon this data analysis method called topological data analysis, which I thought was really useful for identifying the shape of data. And this is what it actually used for, to identify the shapes, the holes, and clusters of data points in a cloud point. I know there are experts in the audience on TDA, but I'm just going to give a brief overview so that everyone can get sort of an idea of how it works. And the idea is, so if you start from the top left corner, you have this set of red dots. And to the human eye, it looks like an inverted 8 or an infinity symbol, just like one big circle attached to a smaller circle. And what we want is an algorithm that outputs some kind of information that says, oh, I see 2 important features. And since we're in a 2D plane, it might seem like there are 2 circles, for instance. And the way we do this is, so we build around each of these red dots a bead that's colored in yellow. And we will increase the radius of these beads at the same time for all of them. And we will observe and take note of when different beads merge to form a new feature. So let's say we start here on the left at t close to 0, each red dot corresponds to a feature. And we start recording the lifetime of each of the beads that is going to grow on the x-axis. And each of the features are stacked on the y-axis. So we start growing the beads. Now, as you can see, some of these beads have merged. which means that the individual red dots that were the previous features have died. And so we stopped recording that on the x-axis. And now we continue growing the beads, and we see that more of them have merged to the point that we can build this structure, this in blue here, based on the previous features. And so this new feature is of an additional dimension. That's why we changed color. And we start recording these their lifetimes on the x-axis again. This is marked in blue here. And then we continue until, say, this little blue circle is submerged, and that marks the end of this feature, and which this corresponds to this little blue line. The big blue circle is still living, and we continue growing the beads, and then until it's all submerged. So we end up having this long blue line, and then we end up having this big blurb of beads, which is represented by the really long red line. So what we obtain here is what is called a persistence barcode. And this can also be read as a persistence diagram, where the death of the features are represented versus their birth. And so this is the kind of information that I was looking for, these two blue dots that represent the existence of these two holes in the 2D plane. But obviously for cages, the idea is to bring that in a 3D space. So I explained briefly how TDA works. How am I going to apply this to cages? So first of all, I was working with two types of cages, metal organic cages and organic cages. And I had two different starting points for them. For metal organic cages, I had no existing data set. I have no reference. And if I did a quick search in conquest for non-polymeric organometallic structures with the 3D coordinates determined, I end up with having 450,000 structures, which is a lot. And I don't really want to calculate the TDA fingerprints on all of them. So the idea is to, similarly to Moths, do some conquest queries and downsize the whole subset. And then with a smaller subset, the idea is to use some unsupervised classification to determine whether a structure is a cage or not. On the organic cages, however, there is some literature on cage data sets. And particularly there's actually only one, which is called the cage database, was developed by Kim Joust at Imperial. And it contains 120 organic cages, and that also contains cyclodextrins and cryptophanes, for instance. And it was very much targeted for xenon-krypton separation. And they were all obtained from a literature review. So I do have some kind of reference. And if I do the same quick search in conquest for organic non-polymeric structures with 3D coordinates determined, I still have a huge number. So similarly as for the metal organic cages, I'm going to use conquest to downsize this. But since we have some data, label data, the idea is to use supervised classification. So I'm going to walk you through just a little bit more detail for the metal organic cages before showing you what I did for organic cages. And as a disclaimer, I just want to say that this is mostly a proof of concept. I just wanted to show that using TDA really can help. And the machine learning part is not optimized, and I am aware of that, and I'm happy to take on any suggestions to improve. So let's start with Moths. So as I said earlier, I started with some conquest queries to downsize this 450,000 number. So these are just some queries that are based on some literature review that I did with Dr. Tanya Ronson from the Department of Chemistry, just to target some specific chemistries. And obviously these queries are not they're not targeted to cages themselves. They're just about the organic, the linkages. And so we will get a mix of cages and non-cages. And that's the point of performing TDA afterwards. So once we have this smaller subset of 3,000 structures, we can extract the coordinates by making sure previously that we're only working with one cage, one molecule, and not like several cages that were in the same file or a cage with different molecules around it. So just one molecule. We extract the coordinates of that, and then we extract the TDA fingerprints. And I actually, before the first time that I tried this process, I just performed machine learning straight away on this. And I realized that a lot of structures that are in a gray area, and what I mean by gray area is that structures that I am not sure if they're cages, and I don't know if anyone knows the answer to that. are included, and that decreases the performance of the machine learning. So what I did is that I removed these structures that I call noisy. So this is one example of a noisy structure. It looks more like a grid. So depending on who you're talking to, it can be considered as a cage or not. So this is very debatable. And so the idea is that using the TDA fingerprints, I can compare the two diagrams using what is called the bottleneck distance and determine whether two diagrams are similar or not. And so I just use this bottleneck distance to discard all these structures that are similar to this one, for instance, and a few others as well. So after removing these noisy structures, I went on to perform the unsupervised classification, and I chose a hierarchical classification. I chose this method because it can output, the results can be output in a format that is called dendrogram, which is what is shown here. And basically, all the structures that are similar are gathered in the same cluster, and different clusters are merged together on like a tree, according to how similar or distant they are. And this is what is indicated on the y-axis. And so what was interesting is that on the first round of hierarchical clustering, the algorithm was able to identify 2D cages specifically, so more like rings, and 3D cages specifically. And I checked them visually, and they were all perfect, either 2D cages or 3D cages. Now, there was still a branch with mixed cages and non-cages. So what I did is that I took this branch and performed hierarchical clustering again. I could isolate some more cages again, but I still ended up having some more mixed clusters. So I performed hierarchical clustering one last time. And this time I had clusters that were small enough so that I can go through them more easily. And I will look at each cluster and just visually check how many of them are cages and not how many of them are not cages. This is not ideal, but having no labeled data set and having not a clear definition of cages, this is the best I thought I could do at this point. So with this, I ended up having 1,400 metal organic cages and I'm about 94% confident that all of that these, I'm confident that 94% of them are cages. So this was the metal organic cages. Now, as I was saying, for the organic cages, we have a different problem because we actually have some labeled data. So I took 929 labeled structures that Dr. Andrew Tarzia from the JUGS computational group gave me. And I complemented this with some non-organic cages examples to form a training data set and extracted the coordinates and the fingerprints. And I used a very basic random forest algorithm, but I thought it was good enough for what I want to do at this point. I had 96% accuracy on the test set. And then I did the same thing that I did for metal organic cages. So I used conquest queries to downsize the initial 400,000 structures to 18,000, extracted the coordinates and calculated fingerprints. And I input that in the random forest to predict the cages from this. So I obtained 7,000 of them. Now, sorry, this was not meant to be here. So basically I ended up having 1,000 metal organic cages and 7,000 organic cages. What we can do next is, for instance, to use hierarchical clustering again to further classify the cages themselves into the different shapes of cages that we have in each data set. And we can then map these information to some molecular simulations, say xenon-krypton separation at room temperature and 10 bars. So I'm not going to show you all the results, but just 2 figures. So here on the left, on each side, you can see box plots that correspond to the different shapes of cages that we identified. So for instance, in the Moths data set, there were 6 in different shapes, and in the organic cages data set, there were about 11. And what was interesting is that, so on the x-axis is the xenon-krypton selectivity on a log scale. And what we're usually interested in is more towards the right. We want to have more selectivity towards xenon. And so what was interesting is that you can see that class 13 has a wide range of selectivities for xenon. And class 4 here for the organic cages has well, not necessarily wider selectivity, but has overall median and average performance in terms of selectivity that is better than the other shapes. And when you look at what these cages are, so class 4 inorganic cages actually corresponds to the CC3 cage that's been previously shown to be the best performing structure focusing on krypton separation on several occasions. And the class 13 over here for metal organic cages is also a similar shape. It's also a tetrahedral cage, but it's a metal organic equivalent with a longer linkers. And so we were basically able to reproduce previous results that were found in the literature and also find a metal organic equivalent to that. So in conclusion, what I want you to take away from this is that, so I've been able to work on two main types of subset, one for extended structures, so Moth subset, and one for discrete molecules, so cage subsets. And for the Moth subset, what I use basically is the polymeric tag plus a bunch of conquest queries. And for the cage subset, I use TDA and some conquest queries. And what I think is pretty nice is that you can see that the polymeric tag and the TDA corresponds to the description of the shape of the structure, whereas the conquest queries correspond to the chemistry. Yeah, so this is the end of my presentation. I'd like to thank my research group and the CCDC and Dr. Tanya Ronson from chemistry and Dr. Andrew Terzia from Imperial for providing me with examples of cages.